I think reverse central dogma. Till that we have done.
Hmm. You can see me, right?
Yes, I can see you.
Yes.
I think we have done till reverse transcription thing, right? The reverse central dogma.
Yes, ma'am.
Okay. Now today we are going to be talking about the packaging of the DNA helix. How does the DNA how is the DNA packed? So basically we know that the distance between the two base pair, it was 0.34 nanometer. That is 0.34 into 10 to the power minus 9 meters. That time, that is the dimension of the distance between two base pairs. And all in all, if you multiply the distance between the two base pair and the total number of base pairs together, you will get the length of the DNA, right? We saw how to calculate the length of the DNA. Now the length of the DNA comes out to be 2.2 meters. And normally the dimension of a nucleus in which the DNA has to be is 10 to the power minus 6 meter, and that's it. So how will that 2.2 meter DNA fit into that tiny, tiny nucleus? So imagine how the DNA must have been packed or, smushed inside so that it can fit inside that tiny space. So in order to fit the DNA, the packaging is extremely necessary. That is why the DNA has to be packaged. So now the packaging of DNA in terms of prokaryotic organisms. Now, even though they don't have nucleus, but still they would require the packaging, or else the DNA will be present everywhere. And that's not what we want.
Yes.
So in prokaryotes, such as the E. coli, the DNA is scattered throughout the cell, right? Normally we know that. But we know that because the prokaryotic organisms, they do not have any kind of nuclear membrane present in them. But we know about the DNA that it is a negatively charged molecule. DNA carries a negative charge on it. Now, DNA, in order to be packaged, it has to be present with some proteins that are positively charged. So that is why it will get attracted and get wrapped around. And then the DNA is packed with some proteins, and it is packed in a region called as nucleoid. So the DNA in the nucleoid is organized into large loops that are held on by proteins. So this is just the very simple kind of packaging that happens in the case of prokaryotic organisms. Clear?
Okay.
But when you talk about the packaging in the case of the eukaryotes, so it's very complex, obviously, because they have a nuclear membrane present. Other than that, they have so many complex proteins present in them. So the positively charged proteins are called as histone proteins. Now remember, in the prokaryotes, there are non-histone proteins. In the eukaryotes, there are histone proteins. Now, histone proteins, they are basic in nature because they are made up of basic amino acids, lysine and arginine. Now, these amino acids, they are positively charged amino acid, and histones, they are organized to form a unit of 8 molecules called as histone octamer. So, 8 histones come together and they form one unit, which we call as histone octamer. Now what will happen? The DNA will start wrapping around this positively charged histone octamer, and then the structure will be called as the nucleosome. Clear?
Yes, ma'am.
Now a typical nucleosome, it contains 200 base pairs of DNA helix. So from the nucleosome, now the chromatin will be formed. Do you know the difference between chromatin and chromatid, if you remember from class 11?
Yes, ma'am. Chromatin is the chromatid is the center, and then chromatin is the one thing, right?
Chromatin is when the nucleus is in a non-dividing state. So the DNA is scattered in the nucleus, and that condition is the chromatin.
Yeah.
But
Yeah.
when the chromatins, they sort of rearrange themselves into chromosomes, right? So that,
That center
yeah.
is called
When that
chromatin.
single unit of a chromosome is the chromatid. So from the nucleosome, the chromatin is formed, and these are thread-like stained bodies that are seen inside the nucleus. The nucleosome arranged inside that chromatin looks like beads on string. Its appearance is like this. Nucleosome. Okay, nucleosome meaning the DNA wrapped around the histone octamer, that condition, right? So firstly, there's histone. Histone comes together and forms the histone octamer. DNA wraps around the histone octamer, right? And then forms the nucleosome. And then the nucleosome gets converted into the chromatin, and the chromatin gets condensed to form the chromosome, and single unit of the chromosome is called as the chromatid. Clear?
Yes, ma'am.
So the packaging of chromatin at higher level requires additional set of proteins, which we call as non-histone chromosomal proteins. Now this is for higher level packaging. For now, we have studied the basic kind of packaging of DNA. Clear?
Yes, ma'am.
Now the chromatin, it is of two types. Depending upon how it is present, how it stains. So the chromatin is euchromatin and heterochromatin. Now, euchromatin is when the chromatin, they are loosely packed. They're not tightly like situated like this. No, they're very sparsely situated. So that is euchromatin. Okay, and they will, you can say, when you stain them, they will stain the lightest. They'll not take up a lot of stain. But when we talk about the heterochromatin, so here the chromatin, they are tightly packed and they will stain darkly. But always remember, the euchromatin region is transcriptionally active, meaning this part of the chromatin will perform the transcription, whereas the heterochromatin will not perform. For the pictorial representation, see here. Heterochromatin, all of them are so closely situated. But in the euchromatin, you see sparse situation, right? Sparse. So that is how, like if you stain them, so if you stain the euchromatin, it will stain very light stain it will get. But here it will stain darker, and because it will stain darker, what happens is that it will be, seen perfectly. But amongst them, which one is more active in terms of transcription? So euchromatin. It doesn't matter how it looks. Now this is the packaging. See, it's very, I love this picture. It's so descriptive. See, DNA. Okay, DNA is here. Now the DNA slowly starts to get wrapped around. Now see, the histones are there. These are histone proteins. The histone proteins, 8 of them join together to form the histone octamer, right? And now the DNA starts wrapping around them. Now the DNA is wrapped around it, right? Now, like this whole entire wrapping and wrapping, it happens, and that structure is called as a nucleosome. Now the nucleosome, from that nucleosome, the chromatin is formed, which is like entangled mass. And then when the chromatin differentiate, it forms the chromosome. Got it?
Okay.
Is it
Yes,
clear?
ma'am. Yes,
This is
ma'am.
a very nice picture. You, like, while you're writing the
Yeah,
notes,
yeah,
you draw
that's true.
this. Okay?
Yes, ma'am.
Because it will like clear out the concepts, and you know that the single units of the chromosome, we call them as chromatids, and they are joined together at the center with the help of
Chromat. Um, chrom. Oh, centromere. Never mind.
centromere. And what is the disc shaped structure on the centromere?
Um, 1 minute. Loci. Huh? No. Wait. Ma'am, can you tell the starting letter?
K.
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 Can you tell the ending letter?
E. Sounds like the name of an ice cream.
Oh. Okay. Oh.
Kinetochore.
trying to cover, yeah.
This
Okay.
much is clear?
Yes, ma'am.
Now let us see the search for the genetic material. Now, we know now that DNA is, well, in fact, our genetic material, but earlier no one knew this, right? So the search started in the year 1926. People performed varieties of experiments to find out that which molecule acts like a genetic material or which molecule gets transmitted from one generation to another generation. So when we talk about the first one, that is transforming principle. Now this experiment, it was given or performed by Frederick Griffith in the year 1928. And he experimented with the bacteria that is Streptococcus pneumoniae. Now see, tell me the shape of the bacteria.
Of what?
Streptococcus bacteria is of which shape?
Um,
Hmm?
I don't.
All 11th you've forgotten, huh?
Ma'am, is it like
Puri 11th, gone. Coccus,
twisted
it's there in the name,
sphere?
coccus.
Yeah, sphere twisted.
Spherical. No, not
Yeah,
twisted, spherical.
like round
Ha,
attached to
round.
each other, right? Like that,
Yes,
twisted
like
and round.
this.
Yeah, yeah. Caterpillar, right? Yeah.
So he performed the experiment with Streptococcus pneumoniae and he saw a miraculous transformation in the bacteria. Now, how did he do it? The bacteria Streptococcus pneumoniae, okay, it is pneumonia causing bacteria, right? It is also known
Hmm.
as pneumonococcus. Now, what happens is that it produces two types of colonies. One is the S colony, the R colony. Okay, these are the strains of bacteria. The S stands for smooth and the R stands for rough. Now, the colony or the strain of the bacteria, that is S, it is smooth and shiny because it has a mucus coat present on it. Okay, and this mucus coat provides this bacterial strain that power to cause the disease.
Hmm.
So the S strain is called to be virulent, meaning it will cause the disease. So it means that it causes severe harmful effects and can even cause death by pneumonia. Okay. So this is
Hmm.
about the S strain. Now the R strain, it is rough because it doesn't have that shiny coat mucus. So it
Hmm.
is non-virulent, will not cause disease. So what Griffith did is that he took the S strain and he injected into a mice. The mice died because it got
Yeah,
pneumonia.
because yeah,
Then he injected the R strain, but the mice did not
mice
die
did not
because
die.
it didn't cause any disease. Now,
Hmm.
what Griffith did is that he killed the bacteria. Now,
The
he
S
killed the S
strain.
strain and the
Hmm.
mice lived because the
Hmm.
S strain was killed. But when
Killed.
he put a mixture of the heat killed S strain along with the R strain, but the mice died.
Mice died.
Right?
Yeah. Hmm.
How? So because
Because
the S strain bacteria, it like and the S strain bacteria was actually recovered from that dead mice. So how
yeah,
did it
because
like
of
how is was there? Huh? Tell me.
the DNA present in the mice, it reacted with the
No.
S
See,
bacteria and it came.
what happened is that even though the S strain had been heat killed, the S strain transformed the R strain and made it virulent. That is
Hmm.
why when the heat killed S strain was given to the mice along with the living R strain, the mice died. So it had been transformed by that heat killed S strain bacteria. So according to him, some
No,
transforming, huh?
can you tell again? Tell again.
See, the first two conditions you understood?
Yeah. This is part I understood. Yeah, this part I understood.
Hmm. So when he took a mixture, okay, took a combination of the heat killed
Yeah. Then
S,
mice dies. Yeah, conclusion. Huh.
conclusion is that basically the reason why the mice got pneumonia, it's because the R strain, which was non-virulent, it had somehow been transformed into becoming virulent by the
Yeah,
heat
that
killed S.
transformation happened because of the DNA present, right?
Yeah, because of the DNA transfer.
Yeah. Huh.
So
Yeah.
according to him, some transforming principle got transferred from the heat-killed S strain to the R strain, which caused the R strain to develop the coating and become virulent. And that is why, and then he concluded, still he did not know that it was DNA. You can't write DNA in this. Okay, don't write DNA because they did
Mm-hmm.
not know. This experiment, Griffith,
Transform.
even though he came to all of these conclusions, but still he did not know that it was the DNA that was causing the transformation. So this must be due to the transfer of genetic material, some genetic material.
Mm.
Then comes the biochemical characterization. Now,
The
this is like
tree.
to give a proof
Exactly.
that,
Yeah.
okay, it was in fact the DNA. So it
Mm-hmm.
was done to find out the nature of the genetic material. Earlier, the genetic material was thought to be proteinaceous, protein, like containing protein, right? Proteinaceous in nature. Then in the year 1933 to 44, Oswald Avery, Colin MacLeod, and Macklin McCarthy, they
Um,
purify. Hmm?
you're stuck in the screen is also stuck.
Is it okay now? Can you hear me now? Can you hear me?
Can you increase your volume?
Is it fine now?
Yeah, no,
So the biochemical characterization of this transform. Yes?
uh, your like voice is breaking. Ma'am,
I
I think
think
the net.
the network on your side is wonky, I think so today. Are you able to hear me, Rida?
Ma'am, hello.
Check your network connection, dear.
Hello.
Check your internet, Rida. Are you able to hear me? Rida, are you able to hear me? Are you able to hear me, Rida?
Can I just switch off my camera? I think there is a.
Do that.
Yeah. Ma'am, can you speak now?
Are you able to hear me?
Yeah, now I'm able to hear.
Hmm. Okay. So we were talking about the biochemical characterization of the transforming principle to find out the actual nature of how it is. So in the year 1933 to 44, Oswald Avery, then Colin MacLeod, Macklin McCarthy, they purified the biochemicals such as protein, DNA, and RNA from the heat-killed S strain.
Hmm.
And they saw that it was the DNA that was causing the transformation. Now it was confirmed that it was the DNA, right?
Okay, so first I should not write that it was because of the DNA, right?
Never write. No. While you're
No.
writing about the transforming principle, do not write DNA anywhere because he did not know. Griffith had no idea.
Yeah, okay.
Hmm. Now, how did they find out that it was DNA? So they applied the protein degrading enzymes, did not affect the transformation. They applied the RNA degrading enzyme, RNases, did not affect. But when they applied the DNA degrading enzyme, that is DNase, then the transformation did not happen. So with this, they concluded that it was the DNA that was causing the whole entire transformation, and hence DNA was the genetic material. Clear?
I have a question. So
Hmm.
for proteases and RNases, the mice, it survived,
Hmm.
but
Yes.
only for DNA, the mice died, correct?
DNAs.
Yeah, DNAs, the
Huh.
mice died. Okay.
See, no, when they were using
Ma'am,
proteases,
your voice is break.
is it fine now? Shall I switch off my video also? Rida, are you able to hear me? Try to rejoin the meeting. Are you able to hear me, Rida?
Hello, ma'am.
Yes, Rida. Able to hear me?
Yes, ma'am.
Hmm.
Okay.
So, yeah, what the question that you asked, actually, see, when they applied proteases, then also the mice died because transformation took place.
Oh,
When they applied RNases, then also the mice died because the transformation took place. But when they applied the DNAs that will degrade the DNA, then the mice lived. Mice did not die then.
yeah. Those molecules, right? The small okay, like they had taken those three test tubes, and in that still proteases was there, still RNA was there, but then DNA, in the DNA, those molecules, I mean, the DNAs, proteins had been removed, like it wasn't there afterwards in the test tube.
Because the DNA got digested. It got
Hmm.
degraded because of DNAs. So ultimately, it was concluded that it was indeed DNA that was the genetic material. Clear?
Yeah.
Now, to give the proof that okay, it is yet DNA only, Alfred Hershey and Martha Chase came into existence in the year 1952. They worked on bacteriophages. You know that bacteriophages are those viruses that infect the
Kill
bacteria.
bacteria. Yeah.
So they took the bacteriophage, and like whenever the bacteriophage attaches onto the bacterial cell, then it discharges its genetic material into the bacterial cell so that when the bacteria will divide, the viral particles will also divide. Viruses are
Hmm.
smart like that. So now
Hmm.
the bacteria manufactures the viral particles. Hershey and Chase, they worked to find out whether it was protein or DNA that entered from like into the bacteria from the viruses.
Hmm.
So they grew the virus on a medium that contained radioactive phosphorus and others on the medium that contained radioactive sulfur. Radioactive, why? Because once you dip anything into radioactive substance, it gets a label. You can
Hmm.
easily distinguish
Explain
which
again.
molecule is radioactive and which molecule is not radioactive. Okay.
But they put the virus in both the radioactive things.
Ha. One was radioactive phosphorus. Okay, why phosphorus and sulfur? Because the virus that grew in the radioactive phosphorus, they will have radioactive DNA because DNA has phosphorus in them. But
Hmm.
the this thing, they will not have the radioactive sulfur because they were not grown in radioactive sulfur. But those that were grown in the radioactive sulfur, they will have radioactive protein, but not the radioactive DNA. Is this clear?
Okay.
Hmm. So now they did the experiment in which 3 steps are there: infection, blending, and centrifugation. So during the infection, both the labeled bacteriophages, meaning one with radioactive phosphorus and the other with radioactive sulfur, they were allowed to infect the bacteria. Then the blending of both of them were done so that the contact, like the viral particle, can go into the bacteria. And then later on, by centrifugation, the viral particles, they were separated from the bacteria. So it was shown that after the centrifugation was done, the bacterial cells, they showed the presence of radioactive phosphorus, because it was obviously the DNA that caused the transformation or the movement from one generation to another because of the new cells. So this proved that the DNA is the genetic material. Clear?
Can you just explain it once more, like just in a recap?
They took the bacteriophages, they
Hmm.
grew the bacteriophages in radioactive sulfur and radioactive
Hmm.
phosphorus. The ones that were grown in radioactive phosphorus, they had radioactive DNA, but they
Hmm.
did not have radioactive protein. But
Hmm.
the ones that were grown in radioactive sulfur, they had radioactive protein, but not the radioactive DNA. So when they caused the
Ah,
infection,
okay, they did. Phosphorus did not have DNA, and sulfur had protein.
no, radioactive phosphorus means that the DNA will be radioactive because DNA has phosphate.
Yeah. Hmm. Okay.
Radioactive sulfur means the protein will be radioactive because protein, they are mostly sulfur, like made up of sulfur containing amino acids.
Okay.
Clear?
Yes, ma'am.
So this proved that, and then they cause the infection, blending, and centrifugation to separate the particles to find out which was it actually. So at the end, after the centrifugation was done, they found out that it was the radioactive DNA was found and not the sulfur. Clear?
Centrifugation is what?
Centrifugation, see, it's a centrifuge is a machine in which you put the suspension and it will like rotate it at a very high speed. And
Okay.
the two different molecules will separate according to their molecular weight. The heavier will settle down, the lighter one will settle up. So when the centrifugation was done, radioactive DNA was actually isolated. Clear?
So they put these two, the phosphorus and the sulfur, in the same thing.
No, they're not the same thing.
No, no, they put it in the same thing, like container.
No, see, they did the centrifugation to separate the virus from the bacteria. Because ultimately the virus particles, they were separated. They wanted to take out the viral particles from the bacteria, not the DNA or RNA. So
Oh,
for separating the bacteria, because bacteria will be heavier, they will settle at the bottom. Viral will be lighter, they will come at the top.
okay.
And after that, it was found that the bacterial cells showed the presence of radioactive DNA in it. Okay.
Okay. Okay.
Hmm. Now the properties of a genetic material. What all things it should have. It should be chemically and structurally stable. It should
Mm.
be able to make its copy, meaning replication can be done.
Mm.
It should have the scope of slow mutation, not fast, not sudden, but slow and steady mutation. And
Mm.
it should be able to express itself in the form of Mendelian characters, right? It should be expressing itself in every generation.
Mm.
But why DNA is preferred over RNA? Because RNA is unstable, right? Because the extra 2' OH of RNA makes it easily altered, that is labile, and easily degradable. And the presence of thymine instead of uracil gives DNA more stability, and RNA has a fast mutation rate as compared to DNA. So which makes RNA unstable. So RNA cannot be taken as the genetic material because it's not feasible. Clear?
Mm.
Now tell
Yeah.
me, what is the last thing that
Okay.
you've written?
Um, uh, 1 minute. Um, uh, um, I have written
I think
Baltimore reverse
okay, this one.
critics.
This whole entire thing is done.
Oh, I'm in the reverse one. Little bit up now. Like, can you just go to reverse central dogma? Yeah. And little bit down. Yeah, this one, this one. Yeah. Now from here you can go down. Yeah.
Hmm? Here, from here only.
Yeah, just up, thoda, like up, and up. up. Yeah, yeah, बस. No, थोड़ा. Yeah, बस. Yeah. Mom, can you go a little bit up? Mom, can you go more up? Mom, can you go down? Can you scroll down? Can you scroll down? Scroll down. Scroll down. Can you scroll down?
Yes, Rida, this much is done.
Red part,
Hmm?
red.
I can't hear you, dear.
Ma'am, just the last part, ma'am.
Okay.
Can you scroll down? Scroll down. Can you scroll down? Can you scroll down?
Done, Rida?
1 minute, ma'am. I'm just finishing it.